So this should be what the webinar people can see right now. And if everybody has read the email, we know the password is workshop to log in. And then I'm gonna, and if everybody, that was weird. Oh, I get it. Okay. All right, so let's start this off. So welcome everyone. So I just wanted to start off giving you an idea. I've talked about this a little bit, but what the history of this workshop has been so far. So we started in 2015, where it was strictly just C-DEBI and C-DEBI adjacent researchers who came and were part of this experience. It went pretty well. I don't think anybody hated me after that point in time. I then ran into some folks from EcoGeo, which was a NSF RCN, who were having a very similar problem in their much larger group of, generally speaking, microbial oceanographers. And they had a very similar problem that C-DEBI was having, which was people wanted to be learning bioinformatics, and there was no easy way to even start them on that path. And so I raised my hand foolishly and said, I'd be happy to do something for you guys. Here's a little thing that I've already put together. And then we then hosted a much bigger event where we actually pulled in scientists and researchers and TAs from all over the country, experts in their various fields. And we got a bunch of people together and we flew them out to Hawaii. And we had 43 people take the workshop then. We also then had 12 presenters. Everybody had their own TA. So even though we've done a very large group of people before, it's not quite the same. So you will see going forward on some of the talks going forward, that there will be names in the corner on the first slide indicating who that person was during the EcoGeo session. So at this point in time, it's been a couple years. The EcoGeo stuff is kind of melded back into the original workshop. And so now we have this like interesting mix of the two. And then I've run it a couple more times for C-DEBI people. And then we are here today in the summer of 2018. And so I just have a short list, so it's about 8 on here, rules for bioinformatics. And so these are some of the rules that I've kind of come up with. They're not all of the rules, but they are the ones that I put some time into thinking about and trying to convey this to people as often and as early as possible. So the first one is that bioinformatics is its own unique branch of science. It's not biology. It's not computer science, it's some mix in between. So if you are starting fresh bioinformatics and you're finding it difficult, that's okay. It's like if you were to start learning some very advanced chemistry or mathematics and you were finding it hard for the first time, it's all right. We're gonna try and work through that here, but don't be expecting to be like, I've been doing biology my entire life. This is gonna be easy. I'm gonna sit down, I'm gonna be figuring it out. I've used computers my entire life. It's gonna be easy, I'm gonna figure it out. It's just not the way it works. It's its own field. It can take some time. Unfortunately, bioinformatics is not binary. So there is no right answer all the time, which is also frustrating for a lot of people. Every step has multiple options. Nowadays, there's lots of groups working on this, lots of different people in different fields working on this. So that means that not only do you have lots of options when it comes to any one particular program at one step, but you also have lots of options of which program you want to use. And that will be, on you as a scientist to kind of look at what's out there in terms of documentation and publications to try and figure out which one is going to be working best for you. Every program out there has publicly available manuals. Some are good, some are bad, some are great. It's always a great place to start. So everything ranges from so one of the programs we're going to use a little later will be Hammer. Hammer has a 76-page manual that explains the theory behind what you're doing, what each step is doing, how it's going to function and how it's going to work. It's really informative. You can get a lot of information out of it. There are other programs out there that the manual is essentially what happens when you get a help message. If you get a help message and it gives you 3 options and you have to figure out what that means. So there's everything in between. And that can be problematic, but we'll see if we can't solve some of that as we go forward. Google has about 90% of your bioinformatics questions solved somewhere. So one of your skills that you will develop as you go through this aspect of science is to learn how to Google your question in a way that's going to bring up the most relevant Stack Overflow response or the manual that you're looking for, or something like that. That last 10% really might not be on the internet. And that's when you have to start contacting people who have written the programs, people who are experts and have maybe done it before, the authors of papers that you see, that's going to be there. If you are Google, or if you're not Googling your question first and you're just emailing somebody, you might be, really pissing them off to start off with. And that's never nice if you actually want them to help you in the future. And so then the last one's here. So a lot of programs will have defaults. Defaults are both useful and dangerous. So obviously a default is something that the person who created the program thinks is an important way to start, right? That's how the data that they work with works best in their program. And that's really nice. But it's not, programs aren't one size fits all. So if your data is something that that person has never thought of before, their default's not going to work for you. That's why there are parameters you can change. There are lots of times when the defaults are going to work fine. There are lots of times when you're going to need to adjust the parameters a little bit to get something that you actually want out of there. Remain skeptical of best answers. Always go back and confirm your results, right? So if you get an answer that looks amazing and it's perfect and it's exactly what you want it to be and it's so revolutionary, it's really good to go back and double check that that's the case, right? So if you're using some type of service that's telling you that you have a gene and it does some function and that's the coolest thing you've ever seen because archaea have never been shown to do photosynthesis before, take a step back. Make sure that that's what you're doing before you go into this deep hole of trying to publish something that you might not have the data that stands back up on the back to support it, right? So there's a lot of reasons why that might not be the case and a lot of reasons why you should go back and take a look at that for a second. There's always something new to learn. This is particularly frustrating for myself, everybody else out there. Every week there are new programs. Every week there are new blog posts. Every week there are new publications that have done something different than the last time that's been done. So this is not a static field. There's no right answer, and it's gonna take some time. And then this last one is. Everybody here maybe has some experience with this, right? And so if you feel like we're skipping something that's really obvious that somebody's taught you already, feel free to let us know, right? There are lots of secrets in bioinformatics that people learn they've given them access to different things. And we want to hear that. So if you have those questions or those secrets, let us know and we'll try and work them in. So this is an overview of what we'll be doing today, sans the group dinner, as explained before. And then tomorrow, there are 4 modules that we'll run through over the course of the day in this order of assembly, binning, annotation, and phylogenetics. And then lastly, as you all know from the links, there's a Google Drive that has a list of all the programs that are installed on the virtual machine. So you can go there. There's links for all of them. So if you want to learn a little bit more about them and or install them on your computer or some other larger computer that you have access to, you can do that. There's a fairly accurate reference and citations list. So for papers, if we were using figures in the PowerPoint, those figures come from papers, and those papers are hopefully referenced accurately in the reference section, as well as these PowerPoints here that I'm going to be presenting on. And then this just came in to my brain right here. One last thing, and I mentioned this with this program install list, is the virtual machine is not really for running real data. A lot of these programs you'll see as we go forward, they do not run on laptops. They will not run in the virtual machine. You'll see in the PowerPoints as we go forward that there'll be gray bars, and gray bars are things that we're all going to do. We're going to type them in, we're going to do that, and then there are going to be black bars. Black bars are things that we had to do at some other data, computer source to generate what we wanted for that input. at that point. And it generally is either because it's very taxing on one computer with only a handful of CPUs, or it's because it took 3 days to run, and we'll let you know what types of data that, what types of time that actually took for what size of data when we can. So there'll be some mix of that going forward. So obviously there's some stuff that even your laptop's not going to do, even if it's not in a virtual machine. And so at that point, you need to find some access to a larger computer. And so there are lots of options for this. Many universities, if you're at a larger university, have access to high performance computing centers. And there'll be people there who can show you how to run these types of programs on some type of large distributed system. If you're a member of C-DEBI, we have a server that people who are actively funded / if I can make a case for why you can be part of it, you can hop on and use our server, which has all these programs and a bunch of other programs that are working. And then there's also options where if your lab has no access to either of those, there's no partnerships that you have that can get you a larger computing source. There's also the Amazon cloud, which we're not going to cover at all. But if you're interested in that, I can direct you towards some blog posts and some other summer workshops that have digital elements to them that will show you how to use the Amazon cloud to spin up a large set of nodes to do some computing power. Obviously, the downside to that is that you pay per minute for what's happening in that type of situation. That's it. So that's the introduction. Does anybody have any questions? I'd say, does anybody want to stand up and give their introduction? But there's just so many of you. It's just going to be a lot. I could do large groups of people, and then we can all kind of figure that out at lunchtime. So the first group I'm going to introduce, because they're here and they're nervous and they're awesome, is our REUs for C-DEBI, who are here, and mostly these two tables right here. If you guys want to raise your hand. So these are our undergrads. I'm making them take this course. Instead of being in the lab and counting cells today, they're here at the course and tomorrow at the course, so I can try and torture them with bioinformatics over the summer. That's going to be fun. We have a group of C-DEBI people who are here at USC, or USC grad students. Those people can raise their hand. It's people like Sarah Hu and some of the other people on the back table. No, guys, you over here. Yeah, we got. Okay, and then we have USC members of the Jed Furman Lab who are in the back right there. So there's 3 of them right there. And then we have 18 of you who have traveled. And so if the 18 of you who have traveled here can raise your hand, that's great. Isn't that, that's everybody. We've covered everybody. All right, so awesome. So that's a lot of people. So with that, let's get started. If everybody can log into their virtual machine, if they haven't done so already. I'm going to make this smaller. Oh, and so I do have one tip for those of us who are, so I've opened up the PowerPoint inside the virtual machine. So that's what's happening right now. It's just loading it up. If you have it situated so that it's outside the virtual machine, where you have your virtual machine running, and then somewhere in Chrome or on your own PowerPoint, it's running in the background. One of the things you can do is you can turn on here under devices. So this is the large VirtualBox command here. You can turn on shared clipboard and make it bi-directional. So that way, when you copy something on your desktop, you can then paste it into your virtual machine. There is, if you're using a Mac, your command keys stop working in that capacity, so you can't do Command C anymore. But you can do right-click paste into the virtual machine. So that's one way of getting around this, if that's how you're doing. If that's how it's set up for you right now. And then I need to hit the present button. I don't need to hit the present button. That is the wrong thing that we want to do. let's do this. Can everybody, I know this is tiny, but I'm hoping a lot of you have this pulled up on your own screen. I'm just trying to share it to make sure that everybody can see it. Otherwise, if I go full screen, we're going to end up with not being able to see a lot of it. Let's see if I can make it any bigger. Does that help anybody a little bit? Okay. Because I'm going to be typing along with you, so I don't want to, at some point it's going to get covered up in the background. All right. So if you have used the Unix or Linux command line before, and these are all things that you know what they are just by looking at, this is going to be a very boring couple hours. I apologize up front, but there are a lot of people in here who do not know what any of these symbols mean, and that's okay. And so that's what our goal is going to be. Our goal is for those people and not for everybody else. So this is a list of some of the very simple commands that we're going to work through, and we'll get an introduction to what they do and how they work and what they'll actually allow us to do when it comes to working with data. And then there's a couple little tools that I introduced at the end that are specifically for working with genomic data. And then we'll move on. And so that'll be it. So the first step we have here, let's introduce the ever-present terminal. So if you haven't yet, you can open a terminal by clicking on this box in your virtual machine right here. And so if everybody's done that, yeah, okay. And you'll get this blinking, flashing purple area. It's very cool with the purple. And so that's going to be our first step. And so what we're going to do from there, you can follow along, this blah, blah, blah. Okay, so the first command we're going to learn is going to be ls. And so this is how we're going to list what's happening, right? So right now we don't know where we are or what's in this command line setup. We're just looking at a blinking line. So we don't actually know what we're going to be doing. So if we hit ls, and this is going to be one of those commands that we're going to use quite a bit, it's going to print out on the line in blue right there, a bunch of a placeholder, where we are, what we're looking at. And so what we're actually looking at here is a bunch of what are called directories. And so this is very similar to a file system, and we'll get a little bit more into this. The file system on your computer, right? So if you have a Mac or you have a Windows, you have these little folders that you click on, and when you click on them, they pop up, and there's a list of other folders and some more files and stuff like that. That's what we've basically just done with the command line, but we've used the command ls, right, to list what's in there. And so one of the things that happens with all these Unix commands is a lot of them have these additional options linked to them, right? And they're called flags. And so the first flag that we can work with is this -a. So if we do ls-a here, right? So it's just a space and then a dash, hit enter. So now we're not only just looking at the things that were there that are easy to look at and the computer wants to show you, right? Which were the original directories. What we're actually looking at is a whole bunch of other secret directories that get created, for managing programs and for double checking. So if you have a program that's running, you'll have things that get installed in the secret area. We're not going to work a lot in the secret area. It's just there to show you that these are what you can do with some of these commands. So, there's more hiding there than you actually think. All right, so we have all these hidden files with the dot. So we could change it, right? So instead of using ls-a, we can do ls-l. And what this is going to do is this is going to give us a long format of the same information that we asked before, which is what directories are present in the current folder that we're working in. And so this here breaks down information about when this directory was created, right? So you can see most of these are older. The last time that they were recreated as part of this virtual machine product. And then we could do things like order it, right? So if we wanted to do ls-lt, we can get the long list format, and the T will order these by time created. And so now you can see that the oldest file generated was this desktop file. So that was one of the first files created when I made it the virtual machine, automatically generated. All the rest of these directories are ones that have been added as the virtual machine has progressed, right? Very simple list. We're going to use list 1,000 times over the next couple of days. So be ready for that. So this is something that we should look. This is what it should look like a little bit on your screen, just for those of you to make sure that we're all in the same spot. Okay, so let's start looking around for things. Look at this. We got a little. Can you go on to the webinar part and answer some questions? They'll get right to you. Kent, sorry. The webinar part is also new for this year. All right, so the next command we're going to learn and also use a bajillion times this next couple days is CD. And CD is short for change directory, right? So you can type in CD, and then our goal here is going to be moving into this directory that's right here called ecogeo, right? And this is where a lot of the programs and data that we're going to work with for the summer. So you're going to do CD space ecogeo, and if you want to be a completionist, you can do a / on that. And then when you hit enter, you'll see that the command line changes. The blinking part with the $ will change a little bit. And so this is to indicate that we've moved into a new directory. This is where we are right now. And so we can actually ask that question of where we are by using the pwd command, which is present working directory. And that gives us just a list of where we are, right? So we're currently in 2 directories that lead to ecogeo. We're in a directory called home, we're in a directory called C-DEBI, and we're in a directory called ecogeo. And so we can do things like in ecogeo, we can list the contents using ls. So we can all hit ls, hit list, and this will give us a list of the directories that are inside the directory ecogeo that we've made it to. We can then do things like move into our newer line set of directories, right? And so you can see a lot of these are named after the things that we're going to do over the next couple of days. Does everybody have, what does everybody's look like? I'm going to come around to the feed. All right. This will not have the chime stuff on it. So we'll have to fix that for the afternoon. Great. Everybody else has a directory that says chime 2 on it, right? Great. Computers, right? What are you going to do? So what we're going to do here is we're going to move into the directory called Unix. So we can do that using CD, and we can type in Unix, and we can hit enter. And again, we can use ls to list the contents here, and we can see that there's one directory there called data. And again, if we go back to pwd, we can now ask where are we on the computer. You can see we've moved from that original location of home C-DEBI ecogeo. We're now in home C-DEBI ecogeo Unix, right? So you can always find out where you are using this pwd command. Okay. So the question is, when did we get into that directory called home, right? And so the home is this landing spot. And so home will be different for most computers that you work on, depending on what you do. So on the server on C-DEBI, I give everybody their own home space to work with, and then they can move to that, and that's where they can keep all of their files. The home for you on your personal computer might be your C drive, where it has all of the different Microsoft containing folders. The home for you on your or in your Mac might be where it lists the other users. It has the downloads folder. It'll be different depending on the computer that you're using. But for this one, home is the Unix home directory. And so we can do something and jump really quickly to that home directory all the way back at the start. So if we just do CD // this is going to take us all the way back along our directory path to the beginning. So this is called the root directory. If we hit ls, this is all of the system directories that are part of the Unix system, right? So you have all these fun, already named, not part of anything that you'll probably play around with too much, number of directories that are running the system, right? You have your sys directory here. You have your mount directory. So if you were to put in a external hard drive, where you keep all your libraries and where you keep all your 64-bit libraries, all sorts of stuff like that. And so for us, this is where everything is stored in a computer, and all the commands that we are going to be running are actually linked through this directory called bin. And so that allows us to type the commands, and the computer looks into bin to see where can I find the commands that you're typing in, and that allows us to get to them really quickly. And so that's just to give you an idea of where the home is. We're going to use this as our navigating starting point. And so to give you an idea of what the structure looks like, it looks something like this, where we have the root directory here with this /, all of these informational directories that keep the computer running, the home directory that we started in that leads to ecogeo, which then leads to all of the directories that we're going to work with, which then also has or C-DEBI, sorry, this is C-DEBI, leads to EcoGeo, and then EcoGeo then has all of the data that we're going to work with for the workshop over the next couple of days. If you want to see a full layout of what the Unix command system looks like, these are the list of all the directories and the subdirectories within those directories that have all this information. This is areas that we're not going to go into. We don't need to know this. We're biologists. We're not going to be playing around with that.
So the webinar people can't see the command line at all. All they see is the opening window of virtual machine. Like, this is what I see on the webinar.
Fascinating.
Yeah.
Great,
that's why
guys.
they were commenting, is they can't see anything that you're referencing.
All right, let's try and change this up. What about this box?
Yeah, now you can see it.
Great. Okay, we'll just catch up everybody on the webinar. Sorry about that. Have to figure that out. Okay, so if we're going to do this, what we need to do now is practice changing directories and listing contents and making sure we get back to where we want to be. So what we can do right now is collectively we can change directory to home. So we can do CD home. We can do CD to C-DEBI. That's a C-DEBI. We can then CD into EcoGeo, and we can CD into Unix. So now we're right back where we started just a second ago. If we hit LS, we can see that the directory data is there. We can change into that data directory. So we can go CD data. We can hit LS there. There's some information there that we'll play around with. And then we can also go all the way back to the root directory, CD/. Okay. So one way we can do this too is we can do this all in one step. And so we can, back in the root directory where we all started, right? So we should all be back here. We can actually, instead of typing out CD, one name, CD, another name, CD, another name, we can actually type out just the destination that we're going to. So we can type out CD home / C-DEBI. Oh, I'm missing a slash. CD/home, EcoGeo, Unix, data. So that can all just be one word. We hit enter, and it moves us all the way to where we wanted to go. So one of the other things that we can do to simplify this, so I'm going to do this. You don't have to do this unless you would like to get a little bit more practice at this. So we can also CD all the way back to the root directory, and we can use tab to autofill the names of the directories or files, which we can do in the future as we go. So you can see this. I will type in CDH. If I hit tab here, it'll auto-complete the rest of home, right? I know where I want to go. Now if I type into C, and hit tab, it'll auto-complete C-DEBI, E, tab, EcoGeo, U into Unix, D into data, right? And so you can use tab to auto-complete all of this information. Again, if I hit LS, this is where we are in data. And so you can do this for file names. We'll do this in a little bit with the file names. The computer is somewhat smart, right? So if you have a bunch of things that have the same name or the same letter, right? So in this case on the screen that we're looking at right here, all of these file names start with the word group. So if I did something, I'm not going to do it. So if I were to do something where I typed in a G and hit tab, it would autocomplete all the way up to the end of group, and then it would stop. And then it would wait for me to tell it what group prefix file I actually want by giving it some more data. So if I typed in a 1 and then hit tab, it would autocomplete that 12.contigs.fasta. But if I typed in a 2, it would wait again because there's an option for a 0 and a 4. And we'll get to play with that in a little bit. The other thing that we can do from here is we can use CD to change directories without giving names. And so we could do a CD ... And so CD with 2 dots after it like that, if we hit enter, moves us up, takes us up directly one directory above where we're working in right now. So from here, we were in Unix data, CD ... We move back up one step, and now we're in Unix. So this allows us to step backwards. So if we hit present working directory, it sees that we're in home C-DEBI EcoGeo Unix. We can step back up one more time, and we can CD back up into EcoGeo. And then if we hit LS, you can see what's around. We can also CD back one more, hit LS, And then there's a directory called Bioinf programs. We can change into that one and list its contents. And this is just to gloat a little bit. These are a bunch of the programs that have been installed on the virtual machine for you. So this is just to make me feel a little bit better. You all see how much work I did getting this ready. Great. Okay, so this is what it looks like, maybe with a little different spacing. All right, so now what we want to do is we want to change back to the directory Unix. So we can do that by going cd ... We can look where we are, and we can do a big group. So we can type in an E and then hit tab. And then we can type Unix and hit tab and hit enter. So we can get back to ecogeo Unix. Everybody needs a working directory for that. Everybody there? There's a lot more squinting in the back row than I would prefer. Let me see if I can make it a little bigger. Nope, that's the wrong way. Is that any easier, back row? Okay, great. Okay, so the next command that we're gonna work with for the Unix command line is this Mcdr Command, which is actually short for make directory. And so what we're going to do is we're going to play around by making our own directories. We're going to delete it. We're going to create some files. We're going to delete those files. We're just going to create a bunch of stuff so we get a feel for what's going on here. So we can do the first thing that we want to do. So for here in Unix, there's our directory called data. We're going to make a new directory. We're going to call it storage. So it's going to be mkdir, make directory. We're going to call it storage. And we're going to hit enter. So now if we hit ls, now there's a directory called storage. Haha, someplace we can store things. And now we can change directories into storage. And if we hit ls, nothing happens because there's nothing in there right now. But we can change that. All right, so we're going to use a command called touch to create a blank and empty file. All right, so this is going to be a file only in name only. There's not going to be any content in it. It's just going to be a place that we could, in theory, be putting text or something like that. And so the command we're going to use for that is called touch. And we're going to create a file that we're going to call temp.txt. So now if we hit ls, now there's a file in there in our storage directory. And we're going to then use the commands copy and move to move this temp.txt around all of the other directories that we have and get an idea of how these two commands work. Because they work, despite being called copy and move, they work very similar to each other and have dire consequences if you don't know how to use them properly. Okay, so the first thing that we're going to do is we are going to copy temp.txt and we're going to make it into a new file. So we're going to do temp or copy and then do temp.txt, right, which I can tab out at any point after I hit the first T. And then we're going to do a space and then we're going to type in new temp.txt. And then if we hit ls here, you can see what we've done is we've taken temp and we've created a new version of it called new temp.txt. And all this is done is taking an exact copy, an exact replica of temp.txt and made a new file that has exactly the same contents. Currently, there are no contents. That's fine, but that's what we've done. We can also do something like we can do copy temp.txt, and then we can do -- like this. And so this is going to copy temp.txt, and it's going to put a copy of the exact same name in the directory above where we're working right now. So if we hit enter, that should work. And then we can go up a directory, right, which we can do by doing cd ... That's going to move us up one. And then if we hit ls right here, temp.txt is right there waiting for us, right? So again, if this had contents in it, it would be moving not just the name of the file, but also those contents into the directory above for us. So this is very similar to if you have your, your computer, you have a file, you hit command copy, you go to a different folder, you hit command paste, you're creating an exact copy of that program and dropping it in there. All right. So then we're going to do something here now. So we want to move back into the storage directory like that using CD storage. So we now have, and then if we hit ls, we have new temp.txt and we have temp.txt. And then we're going to use the move command, which is mv. And so we'll see, you can see something strange is going to happen with that. So if we do move new temp.txt, and then we can tab that out, and then we write old temp.txt as the name of the file that we're going to be moving it to, and hit enter. Now if we hit ls, you'll see instead of copying new temp text and having new temp text existing and a new file called old temp.txt, what we've done is completely replace new temp.txt. It's now old temp.txt. So if there was contents in here, they'd be the exact same contents, but we've destroyed the original and created a new copy of it. And so move is one of these programs that's really nice, but very dangerous. So if you move something and you put it into a file or directory that either you can't read or write to, or you name it improperly, it's going to go missing and you're going to have to spend some time finding it. So it can be a little difficult to do. But it also works like this, where if we do move old temp.txt, and in this case, we could type out a specific location where we want to move it to, right? So I want to use the full, so one way we could do this is we could, let's just do it the way it's on the screen. I don't want to confuse anybody. So we can say specifically move it to home, C-DEBI, eco, geo, Unix data, right? So if you remember correctly in the file structure, Unix had one file originally called data, and then we created a new one called storage. So all we're doing is we're taking it from storage, moving it up, and then putting it into data over here. So if we hit enter and hit ls, you can see unlike copy, which left a copy of new temp text in the folder we were working with, we've literally moved the file from one directory to the other. So now if we cd up from storage and cd into data and hit ls, you can see now we have old temp.txt hanging out there. Cool. So just to summarize here, copy will make a copy of a file and we can use to move a copy of files to other directories. Move destroys the original and places all of the contents into either a new file or a new directory. Okay, does that make sense? Everybody got some head nods? Great. Okay. All right. So we want to look at where we are. So we're going to hit ls if we haven't just done that in eco, geo, Unix data. You can hit ls. You have all the information there. So now we're going to work with the remove command. And so remove is a great way of losing all of your data if you do it improperly. Remove, unlike, I'm going to say this, and there's a little asterisk in the end of it. Remove deletes your files, and there's no way of getting them back. So it's very one of those things. Do not delete something unless you want it to be deleted forever. There is a little asterisk. Sometimes it can be saved. That's not all the times, but sometimes it can be recovered if you're lucky. And so right now we have old temp.txt sitting right there, and it's got nothing in it. And this is the data folder. We don't want it here anymore. We got to get rid of it. Okay, so we can use remove, so rm, like this, and then we can do old temp.txt. We can tab it out because it's the only O really easily. We can hit enter. And then when we hit ls, it's no longer there. So we've been able to remove the file. So what happens though if you say hypothetically we're running a workshop for bioinformatics and you had told everybody to create a directory called storage, but really there's nothing in storage and we wanted to delete storage? How could we get rid of the directory storage? So what we can do is we can move up a level. We can move into storage itself. We can get rid of temp.txt there, but then we still have this directory floating around that's empty. So we've done that. There's no files in here anymore. We can move back up to the Unix level, and then we can get rid of storage. And we can do that by doing remove and then using the flag -r. So -r is short for recursive. All it says is that, so normally, I can show this command to everybody what it looks like. So if I just do remove and hit storage like this, it says you cannot remove storage as a directory. It's trying to save you from yourself, right? That's what's happening right here. Because removing a directory would delete everything that's in there along with it, right? So you don't want to be able to do that willy-nilly. You got to think about it. So they make you type in 2 extra letters to make sure that that's what you really want to do. So if I do remove -r storage and hit enter, that's it. It's gone. If there were all of your data files were in that directory, they're gone now too. If you had a super awesome data structure where you had lots of different directories with all sorts of information and it went really deep and you had, that's all gone too, right? So just be careful with your remove -rs that, again, this is something that you want to remove and have gone from your system and your computer. Right, now to stop scaring you. So this is a little like, let's do it together kind of thing. So what we're going to do is in Unix, we're going to create a directory called the best directory ever. We're going to go into that directory. We're going to make a file called glam.txt. We're going to change glam.txt to former glam.txt. However you want to do it, you can do that with copy. You can do that with move. We're going to remove it. We're going to change our directory back up to Unix. And then we're going to delete everything that we just did by removing the directory best directory ever. So this is a little test for you guys to get some experience just doing it with like a list of words here. If you have questions, we'll all go through this together, but I'm going to give you a minute to step back and do all that. I'm going to wander around. If you have questions, sorry webinar people for not having the first 10 slides available to you. It was a good planning on my part. And please raise your hand if you have problems.
Bruce, like, really tough. I watched your stuff.
Right. So like, yeah, so like nano.
Yeah. Is it the next one that they're expanding the field?
I'm not sure. So you made the file, and there's contents inside of it. What it is. Right. Yeah. I don't know.
Oh yeah, or the one after. Yeah.
Yeah. Right.
Yeah. That is true. There's an evolution.
Yeah, which is at my desk, I have a little card that I have written down on all my like. I don't like the source. So that's what I was thinking. So yeah, like just. So yeah, I did try.
Understand why. No, I mean, I understand. I don't understand why programs have a computer where it is just.
I've never seen this area as a job or something.
Like, what's the? There's something there.
I was kind of bizarre. Everybody now seems to be the expert in Python.
So I emailed you yesterday and told you I was looking at errors when installing.
Yeah, I know.
Google this error.
It's like that's because like that's what the other people. I'm just like,
But you have to
I guess I'm gonna be better job.
right.
Like it's just not even.
Not that I know of, but maybe.
No, you probably if you haven't done it yourself.
No. I mean, I've.
Especially for
Yeah.
All right, so there's a lot of murmuring. Is that murmuring because we're all talking about what we're doing, or murmuring because we've successfully done it? Success on this table. How's this table looking? REU table. Yeah, great. It's a nice shorthand for you guys. Sorry. Awesome. All right, so that was a little, fun to just do it yourself. Everybody have a really good time. Some cool names, glam. Anyway, all right. No one has to like it. That's fine. You're all here. All right, so if everybody has done this, we can move on to the next slide. Okay, so we should be able to change into our data directory. We can list our contents. If we haven't, we can remove old temp.txt, but I think we have already done that already. And so now we can actually look at some files that look like biology. And that's only if you count looking at DNA as biology, but there you go. So in this directory right now, we have 3 files that end in the suffix FASTA. And so this is a specific format for looking at molecular data, and it has a specific format for how it functions, right? And so it's summarized here on the slide here. What we have normally in a FASTA file like this, or a FASTA file, is we have a caret that represents the header line, and this header line then has information on it. Everything before the first space is known as the ID, and then everything after that is known as the description. And you can put a lot of information in here. If anybody has gone and looked at, I'm trying to think off the top of my head, what contigs from the GOS data set, which maybe has we've aged out of that at this point for some of the grad students in here who have not GOS. Why would we do that? There's only 10,000 reads in there or something like that. But they store all of the information in the header. It's like 7 lines long. You have an ID, you have its sequencing platform, you have when it was collected, the day it was collected, the time it was collected, the depth it was collected at, all the information in there. And you could just keep adding information. There are lots of programs that hate that, but you can add a lot of information to the header. The next line, anytime you hit the enter, you now go into the actual data side of it. In this case, this is an example of DNA. And so you can have 2 different structures here where the molecular data is stored either as one long line and it will just keep going, or you can have it where it's structured that after every 80 base pairs or every 80 amino acids, there's a hard enter, and then it will go to the next line. And so when you are running these programs that are looking for this type of information, they're looking for the header, and then everything after the header for whatever the next period of time is data, and then it looks for the next header to find the next sequence that it's looking at. So we can actually look at these types of files and get in over our heads a little bit. So get it? Head? The next command is head. Anyway. Head is a command that you can run that will display the first 10 lines of a file. So we can do head and enter the file name, and it'll give us 10 lines. And we can use tail, which is the opposite of head, to give us the last 10 lines of a file. So we can do that. And I think we do that on the next page, because one of the things, and I'm going to show this as an example. So because of the way I just described it to you, right? A FASTA file has a header line, and then it can have all of its data stored as one long line until the next header line. So if I hit head on one of these contigs FASTA files, and there's a lot of sequence data there, it's going to print it all out. So you don't have to do this, but I'm going to do it, and you'll see what that looks like. So this is only going to be the first sequence, 10 sequences of this file, but because there's no hard enter after the first line of information, it's going to keep spitting it all out. So it does look like that. And so what I've only done is printed out the first 10 lines. They just happen to be very long lines, and it can do, and your computer can do stuff like that. It's not always useful, but it can do stuff like that. Same thing will happen with tail. So if I do group 12, it's printed the last 10 lines. You can't tell because it's all up above there. So what we can do is use the command less. And so less allows us to do something like, so you can do less, and everybody can do this. So less, when you do less group 12 contigs.fasta and hit enter, it brings up a screen inside your Unix command line that you then can scroll through, right? So you can see the first name, the first header of the first sequence that we're working with is contig_2. There's a whole bunch of DNA data. But then if we hit the down arrow, we can start tracking through this information. So you're going to go through a lot of information here for a while. If we want to do this a little faster, we can hit the space bar, and this will advance a whole page at a time. So it's going to be hard to see because it's DNA, but you can see it jumps, big jumps like this, and we can keep doing that. I think this is a 20,000 base pair contig. So eventually we'll hit the next contig name. Oh, that's not it. Here somewhere. I quit. All right, so we've used spacebar to go down a whole bunch. We then can use the B button to go reverse back. So instead of, oh, we just saw it. See it? There it is, right? So you can use spacebar to go down, and you can use B. So spacebar goes down a whole page, but you can also use B to go up a whole page. So you can go back up just as quickly as you went down. And you can hold it down, and it'll go forever. And everybody will go, that's a lot of A's and T's. And then you get to the top, and then we can hit the Q button to quit out of less. And it takes us right back to our command line. Okay, so I showed this, so I don't want you guys to all have to go through this because we're going to get into a little bit more detail about how we can get specific information we want out of this. But you could, in theory, if you were eager to, is use head to display the first 10 lines of the group 12 contigs, or the first 5 lines, or the last 10 lines, or the last 5 lines. And so to do a specific number like 5, you can actually tell it that. So I can do head-5, and that will give you the exact number of lines that you want. So this should end, this should end on, because 5 is odd, and we're alternating DNA versus header. The last line here is a header line. But you can change that to however you want. Oh, that's not what I want to do. So you do head-25, and this will do the first 25 lines of group 12. It'll print for a while, and then again, because it's odd, we'll see a header there like that. Okay. All right. So that's not really that useful, right? I don't want to hit head and get all of the sequence data there as well. What can we do to actually get something out of all of that effort? So we're going to use grep, which is a command that allows you to search a file for a specific pattern, and then WC, which is short for word count. And word count allows us to count the number of words, lines, and characters in a file, which can be useful if we're working with something like, big set of DNA, and we want to know how many DNA letters are in a file that we're working with. So we can do this all together. All right, so the first thing that we're going to do is we're going to use Grep, and we're going to use the thing that distinguishes header lines from non-header lines, which is this greater than sign or caret. And the thing that we want to be really careful for, and I can already see it here. So in the, right here, if you're going to copy and paste this, this is not going to work. And it's not going to work because PowerPoint has made the smart quotes around it. So we want to be careful of that. The other thing that we have to do is using a caret can be a little dangerous, right? So we're going to talk about this in a little bit. Carets are also ways of redirecting information into a new file. So when we're using Grep, we want to make sure that we use quotes to denote the fact that we're not trying to write a new file here. So we can use something like Grep, open quotes, caret, close quotes, group 12, like that. And this is going to find all of the matches that match that caret. So if you hit enter, it's going to print a list like that. In our nice version of the virtual machine Unix, we actually can see that it highlights the little caret to show what it's been matching on. That's great. If you scroll up, you can see it's a pretty long list of numbers counting backwards and going and going and going. But that's not that useful. Like, what are we going to do with that? All right, that's fine. I hear you. Don't worry about it. So what we can do is we can do either something like this, where we can hit Grep, open quotes, caret, close quotes, group 12. We can then use the symbol called a pipe. So this is the command that, or the key that you never use. It's right above the backslash. The shift, it will create a straight line like this. This is called a pipe. And what pipes do, it allows you to string together multiple commands, right? So we're going to take our grep command and we're going to string it with our word count command. And this is going to do something that's very fun. It's going to give us a bunch of information. And the way this breaks down is the first line, or the first number here is the number of lines in the file, and then the number of lines that we detected. And then this is the number of letters on total in all the lines, right? So if you were to count up all of the config underscores and numbers, how many actual characters were generated there are there as well. So this tells us in this config file that we have 132 sequences, right? So it's a nice, easy, quick shortcut to figure out how many sequences we have in there. Yep.
Can you say again what the difference between the first one and the second one is?
Yeah, I'm having a brain fart about that right now. It will come up in a second, and I'll get to it. But I'm having a brain fart right now.
What if you have zeros that come up instead?
You have zeros.
Like 3 zeros.
So you didn't. There's nothing. So that's saying that it's detecting nothing right now.
Did you type the money symbol before the grep? I did the same thing. So I deleted the money symbol, and then I got not zeros.
Here, Elena's going to help you.
Oh, and ninja tip, if you hit up, you can.
Yes. Yep. And so one of the things, so this is just, if you hit up, you can get the command that you just worked with, right? So if we wanted to shortcut to this, right? So right now, this is what we just typed in. So hypothetically, we could go back to what we typed in before, where we got this nice long list. And instead of having to type it all back out again, you can do pipe word count, and that will generate that again. Okay. Next. Yeah, okay. So we can do this now with the other files that we have in the folder, right? So we can do this a couple ways. Let me show you a couple ways to do this. So we can do grep, open carets, open quotes, caret, close quotes, group 20, right? And we want to know how many contigs are in there. We can do word count. And we can do word count -L, which will just give us the number of lines directly. So there's 203 in that particular folder. Or if you are wanting to use the up, if you hit up, you can go back up to where it was group 12. You can delete the 12 and replace it with a 20 and generate something that looks like that as well.
What was the -L after?
So -L will just give you the number of lines. So we can do this for other things that might be matching, that we might need to match, right? Not just for carrots. So one of the examples I have up here is, what if we asked how many headers have the number 47 in them? And we know we're going to hit headers because DNA doesn't have numbers in it. So that's pretty convenient. And so what we can do is we can do something like grep, open quotes, 47, close quotes, group 12. And we can put word count or word count dash line and ask, well, how many headers have the word number 47 in it? And there are 7. And we can change it again. So if we hit up like this and bring it back up and scroll over, we can put an underscore in front of 47. And so this is going to particularly find matches where the 47 is at the start of the number as opposed to the end of the number, right? Because the structure that we're working with has contig underscore some number. So if we hit that, you can see there's only 3 that have the 47. So this would cover things that are the number 47, the number 470, and 4,732, right? Would be examples of these things that we could find with that. As opposed to 47 by itself, where we'd see 47, we would see 347, we would see 3,047,1 or 3,000,004, wherever the number 47 shows up. So you can play around with this and find the elements that you want as you go forward. Okay, so now we're going to play with fire a little bit, and we're going to use the command. So we're going to use grep to use the caret to grab a caret and look for the header lines. And then we're going to use a caret to direct that information that we're getting into a new file, right? And so we're going to do this just like we've been doing before. We're going to do open quotes, caret, close quotes, group 12. So this is going to catch all of the header lines. And then we're going to redirect it using a caret by itself. This is very important, caret by itself. And we're putting it in a file that we're going to call group 12 underscore IDs. And so this is going to store all of our ID information in a file, something that we can go to. So we can hit enter, right? And we can play around with this a little bit. So if you wanted to look into this file, we could use less, group 12 underscore IDs. You can hit enter. It's now something that you can scroll through and you can see what's there. You can look for the 47s by hand if we wanted to. You can hit Q to get back out of that. And now we have a file. If we hit ls, it's here. So we have group 12 contigs.fasta, and now we have a new file called group 12 underscore IDs. All right, and so we could do the same thing with the 47 example. So we do grep, open quotes 47, close quotes, group 12 contigs.fasta. We can redirect it into a file called group 12 underscore IDs underscore with underscore 47. So this is going to create another new file. We can hit ls. We can look at this file using less here, group 12 IDs with 47. If we hit enter, you can see here are 7 candidates with 47 in the name. Great. Anybody with questions on that? Because we're going to keep playing around with grep for a while.
How would you indicate that you're just looking for the contig underscore 47 if you're looking one? So have the grep stop 47 and not search for anything else thereafter?
Sure. The first example, like you're saying, the first match of it, you want contig underscore 47 and that's it by itself. There is a flag for that. I can get back to you on that one. Yeah. Okay. Next step. All right. So now what we want to do is we want to play around with the command cat, which is short for concatenate, right? Which is the idea that you're going to take something and you're going to just stick it on the bottom of the next thing. And so we're going to play around with this. So the first thing that we can do with cat is if you use cat and you don't put, so we're going to use this group 12 IDs with 47, and you do it by itself, all by itself, all it's going to do is it's going to print the contents that's in the file. So we can hit enter. And so this is our file that we just looked at. It has all the 47s, all examples of 47. There should be 7 there, right? So that's listed it for us. So if we do cat with a caret like that, right? So carets create new files. So instead of printing it to the screen, we can take everything that was going to be printed to the screen and we can direct it into a file with a new name. And so we can do that and do temp1 underscore IDs. And if we do that, hit enter and hit ls, you can see there's a new file called temp1 underscore IDs. And so if we wanted to do this, we could also do copy group 12 IDs with 47. And without putting a caret, we can put temp2 underscore IDs. And hit ls. So what we've done is essentially given an example of how you can do the same thing just using different commands, right? So cat with a caret into a new file will just take everything that's in one file and put it into a new file with a new name. And like we learned before with copy, you can take copy, the name of a file, give it a new name, and it will take all the contents in that original file and move it into a new location. So right now, if we're lucky, you should be able to go into ls temp1 and take a look, and it should list 7 IDs. You can hit Q to get out of that. And then if you do less temp2, the same 7 IDs. So they're exactly identical to each other. 2 different ways of doing it. So now we can do things like take 2 files and stick them together, like the name concatenate says that it should be able to do. So we can do something like cat temp1 space temp2, just like that. We can use the caret to redirect this output into a new file, which we can call duplicate, because there's the same, they contain the same information, duplicate IDs. Now if you hit ls, it should be there up here at the start of the alphabet. And if you hit ls or less and go into it, you can see what it's done is you have your first line here of 247, goes all the way up to 13,047.1. And then you can see it repeats, right? So it's just taking the exact same, because they were identical to each other, it's taken everything that was here and just stuck it on the bottom. And now you have a line or a file that has 14 lines in it. Great. So now we could do this again, where we're going to do something where we do group. So we're going to use grep, open quotes, 51. So this is a new number that we're going to work with. And we're going to pull this out of group 20, right? So now we're going to have a list of IDs from a different source in a new file. So we can do that with something like group 20 underscore IDs with underscore 51. So you can do it that way. And so then we could do, so this could be done a couple different ways here. So we're going to work through a couple different ways that we can do this. So now if we wanted to take our duplicate IDs file and add in all of those 51s, we can do that. And we can create a new file called multiple IDs. So let me give you a couple examples of how we could do this. One, we could do this. We can do cat, duplicate group 20 IDs with 51. You can put a caret. We can type in multiple IDs, and we can have a new file that's going to contain 14 lines of file or header names that have the 47 in it, and then another bunch of lines that have the 51 in it. We could also do something like this, where I could go back to this step and do this grep part again. And instead of directing it into the file here, I could pipe it to cat and pipe cat with duplicate and then put in multiple, right? So there's a bunch of different ways. At this point, we're starting to get a lot of little tools here from the Unix side of things where you can start mixing and matching what we're learning and do it in lots of different ways. So this way, if we did it this way, would not generate a new file. We wouldn't have a file called group 20 IDs with 51, you would skip that step entirely and just go straight to creating a slightly longer file named multiple IDs, right? So there's lots of ways to do this. I'm going to go back the other way and do it the way that we did, or was suggesting the first time, where we're going to do duplicate. We're going to go 20 IDs, and then we're going to call it multiple underscore IDs. All right. So now we could do things using the unique command and the sort command, right? Because now we have a file that has a whole bunch of stuff in it that we may not want to be there, right? So we can, unique obviously is going to work in a situation that you hope it, based off the name, is going to find things that are duplicates of each other in a file and tell you where they are. Sort is going to be very similar in the same way, where your hope is going to work the same way, where you can be able to take the file that you're working with and actually sort them so that they're all in an order some way. I'm going to tell you right now, these new two commands never work the way that you want them to work the first time. So that's why we're going to go through them a little bit. They take a little bit of finagling to get into the right spot to do what we want to do. Okay, so let's take a look. Let's take a look at less multiple IDs. So we have the 247 as the indicator at the start of the first file. Then we have another 247, which is its duplicate further down. And then once we get to a certain point, we should start being able to see 51s everywhere, right? So there's a whole bunch of 51s. And so when we're done looking at this, we can hit Q to get out of it. And so I'm going to hit unique, multiple IDs like that. And I'm going to hit enter, and it's going to print it to the screen. And we're going to get rid of all of those duplicates. And if we scroll back up, you'll see we failed at that completely miserably. There's 247 right here, another 247 duplicate. It didn't do it at all. Come on, guys, why? The reason is because unique works in a way where it's going to look at it only looks at the line directly below it. So if you have 247 is the target that you're looking at, and there's another 247, it will only find it and tell you about it if they're right next to each other. Great, thanks Linux for being super awesome about this. So the way we can do that is we can use sort, multiple IDs. We can use a pipe. We can type in unique here. I'm going to put a caret because we're going to take this new information and we're going to put it into a file called clean IDs, right? So it doesn't have any duplicates anymore. And now when I hit enter, and we can go look, so we look here, you can see that we have clean IDs up here. They're all right there. If we take a look into it, clean IDs. All right. So if we look for 247, are there two 247s? The answer is no. Yay. Great. We got rid of the duplicates. You should also take note here of the way this is sorted, right? So it's not sorted based off of, because, okay, if this were just numbers, we'd be fine. But this is numbers and text. And so what it does is it looks for all the text first to see if it can sort it alphabetically. It can't, so because we have contig through underscore all identical. And then it just looks at the digits and tries to sort it by the digits. Like it was actually, these were A, B's, and C's, is trying to sort it that way. So what you have is you have all the ones in this first digit ordered, followed by all the numbers then in the next. So you can see here, it's easier with these 3 digits here, that 247 comes before 447, which comes before 476. But then up here, you can see that 10,000 or 10,051 comes before 13,471, which is great. But then it also, those all come before 1,651. So, there are ways around this, but that's just to give you an idea of if you're just using sort in a raw form, that it will give you an output that you're not exactly expecting, but is still very useful. Okay. All right. So let's do a little bit of cleanup here. And so we can do this a little quickly. So we want to get rid of those temp files that we created before. We're going to use the remove command. We're going to use temp. And here we're going to use, so right now, right, there are 2 temp files. One is temp1 and one is temp2 underscore IDs. What we can use here is a star, right? So this is an asterisk to act as a wildcard. And so what this is telling the Unix command line is remove everything that starts with temp and then anything after that, right? So we could have anything after this word temp that would be here. It's going to find everything that starts with temp and delete it all. So if we hit remove star and hit enter and hit ls, both of those temp files have disappeared now, right? And so we can do this with a lot. We're not going to, we're not going to spend a lot of time. At the end, if we want to, we can burn it all down and we can do like, remove group and then do all that. But we're going to, and figure out different ways of deleting the things that we want to delete. But we're not going to do that right now. What we are going to do is we're going to use 2 different flags in unique to try and figure out if there's something that we can do to get more information for what we wanted, right? So we're going to do this first. So if you just follow along, and I'll tell you why we're doing it. So we're going to do sort multiple IDs. We're going to pipe it through unique, put a -d. We're going to put a caret, temp1 underscore IDs. Hit enter. So that's going to be 1 file. And then we're going to hit up. You can go back and we're going to change that 1 to a 2. Make sure you change the 1 to the 2. Otherwise, we're just going to create 2 files of the same name. And then you're going to change that D to a U. And we'll have 2 different files that are generated from this. Okay. And then if you look at them, less temp1, maybe we can figure out what these flags have done. So remember, multiple had 2 copies of everything that had a 47 in it and copies of things that had a 51 in it. And now we've used -d and we've only had 47s that are left. So what has this done for us? I know there's not been a question and answer before at this point, but so what it's done is it's found everything in that file that was a duplicate and kept it and thrown everything else that was unique and thrown it away. So the same thing, if we look at temp2, does anybody want to take a guess at what the -u might stand for? Keep unique. And so now is what it's done. So it's found everything that was a duplicate and thrown those away and is now keeping everything that's a 51. So now we have 2 temp files that have gone back to our original files of group 12 with 47 and group 20 with 51, right? So we've replicated those original files now, but having had them all duplicated and cleaned up. Right, so sort, right? So we looked at it for the duplicates, we looked at the uniques, and now they're equal to these original files. All right, we're getting there, guys. We're going through. We're getting through this. We're getting through it. It's been an hour and 20 minutes. How does everybody feel? Yeah, good, okay. You're not standing, just remember that. All right, so our next command we're gonna use is sed. And so sed works in a similar way to grep, right? We're gonna use its ability to look for pattern recognition to do something to files. But what it's gonna do is gonna allow us to change things as a big group, right? So we can take something that we know about our file and change everything in it all at one time. Right. So what we're going to do here is using our file clean IDs. So let's take a look at what those look like. So with clean IDs, you can look at them. I'm going to actually post them up like this. So clean IDs, you got some contigs and stuff like that. If I do head-5 clean IDs, right, it's going to give us the first 5. So it's something that we can reference right here. All right. So what we want to do is we want to take sed, and then this is a single quote, open single quote, lowercase s / big C, capital C, / little c, close quotes. Oh, sorry, there's a slash there. / close quotes. Clean IDs. Oh, there's no space. Missing a space. There we go. Okay. So now if we hit enter, so this is going to get print out to the screen. We should see something. So, right. So if this is what these look like right now, I hit enter, and I've changed, it's magic, I know. I've changed all of those big C's to little c's. It's great, right? Great. So we can do this with all sorts of different commands or all different patterns that we might want to find. So if I hit up here and change the big C to an underscore change the little c to a period, and hit enter. So now it's printing out, and you can see that all those underscores have become periods, and that's great. But you can also see that the C's have gone back from being lowercase c to capital C. And so the reason for that is because we didn't actually make a change to the file. All we did was we asked the computer to do this change for us, but it didn't save it anywhere, right? We were just running it and it was printing it out to the command line, right? So that first change that we made, where we made a big C into a little c, didn't get stored anywhere. And the second change that we made just went back to the original file, kept the big C the way it was, and removed the underscore and put a period there. And again, this has happened here where we actually haven't stored this information anywhere. So it's just printed to the screen and we can visualize it.
Assuming you wanted to make multiple changes at once, let's say change all the big C's to little c's and also all of the underscores to periods, would you just string all of the
It's not as direct as that. There are ways you can do that. It gets a lot fancier to do things like that. My recommendation for something like that, unless you want to go really deep into how sed works, is you can just do it in a multi-step fashion. Unless you're getting really like fancy and there's like a ton of stuff that you want to do, then you can look into it and find a way to do that. So the last thing that we want to do, and this is something that we actually do want to save. So if we hit up and we have this command here, the underscore period command, we can hit a space, we can do a caret, we're going to call a file called new clean underscore IDs, and then we're going to scroll back along and we're going to delete the period and we're going to change the underscore to a caret. And so what this is going to do is going to go into our file, which is a bunch of header lines with carets in them, just like right here, shown right here. And we're going to find all the carets and we're going to remove them and we're going to put them in a new file. So if we hit enter, look at this one. If we look at new clean IDs, so now we have config underscore number with no header line, no caret there to begin with. And this is useful. This actually is one of those commands that you end up kind of using a lot once you know how to do it, because it allows you to create a list of IDs that don't have carets in it very quickly. And there's a lot of programs that want your IDs in a list, but they don't want the caret. So this allows you to get through that and generate that list fairly quickly. So we take a second here because now we're going to play around with a program. Does anybody have any questions about this? No. Everybody's all caught up? Yes? Cool. All right.
No, I've done something.
Oh no. Okay, so this is just so this is because you didn't close coordinate right here. And so we can actually, so what had happened is, so you went into sed, and sed wanted to then like have this interactive version of it with you, which was not very useful for what we wanted to do. And so then I just stopped the command from running by using Control-C. So we're actually flying through this. So this is a great time if people have questions. There's not a ton more left to do going forward. We have lots of discussions after this, but it just will mean that there'll be a long lunch break. And that's exciting, right? Something for everybody to catch up on to get ready for the afternoon, which is filled with stuff. And then I can fix the virtual machine that I'm working with to make sure that I have the right one. For some reason I don't, which is great. And we can make sure that's all working fine. All right. People getting coffee. Let's take a formal break here. Everybody can get some coffee, go to the bathroom. There's like a piecemeal here. We'll just do a formal break. And then we'll come back in 5 to 10 minutes. How's that sound? Thank you. Instead of half talking about it.
okay if I don't have all the same like content ID files that you made, unique stuff with the.
Yes.
Are we gonna continue?
Do you have multiple?
Stuff? No, because my micro 12 is just like not thinking. So I didn't do it.
So we always have like 72.
So, yeah.
We'll just follow on. And then I can do the files in the
New
practice.
directory.
These are,
Check the files that I made.
these are.
Start over.
Okay, great. We do play around a little bit, but it's only a minute long.
Joel and I did this like that several times. It's X7 and try to figure out
Yeah, that's it.
wait, yeah, pretty sure. So, never worry about it. I think times 10.
Theoretically, it's already installed. We'll find out whether that's the case or not.
This is why.
Yeah, Google how to convert X to Y, and then try that. Sometimes it works. Sometimes it works. And at least with this.
But we're not.
Because if I can like figure it out, I'm not going to figure it out. I'm not going to spend the amount of time.
That's pretty much how it was the entire time I traveled.
I mean, it hasn't. It's been saying. I mean, I think.
Yeah. Yeah,
I'll go to sleep.
I think anyone wireless thinking about this have the combination of the notepad. Yeah, and I'm typing in plus the PowerPoints. So I take this classes as well.
Yeah, I do it in Word.
Computer is not very good, so it sometimes takes it like 5 to 10 minutes to load Word. doesn't take it any time to load Notepad. So that was the reason it started, and now it's just become a habit.
Yeah. So let's come back.
Probably, but this is, it works. This is what I know about you. It would just be riddled with typos for me. It is riddled with typos. So one of the things that I do when I get back to class, I go through my notes, make it intelligible what it was that I was supposed to, what it was that I was talking about. I used to do that. Yeah.
I can't see the questions. It only goes to me.
Okay.
Like when I was on the level,
Okay.
I can't see any of the questions.
Read it.
I've only had that one question.
Yeah, I figured, because it should be popping up. So I started seeing it back, so I can just like do better.
I can, for the next session, before we start, I'll give you.
So I think that's what it is.
Okay.
Hey, let me ask you a question.
Yeah.
So can you make the terminal window just a little bit bigger in comparison to the PowerPoint?
Sure.
So that if you get behind, you can see some of the stuff you've done
Yes,
previously?
I can do that.
Okay, thank you.
I don't know.
Not stereo.
Oh, okay.
Minnesota has the most fresh water in the state.
They do have a bit of spare.
It's actually the best.
Webinar people, we are waiting on 2 more folks to come back, and then we'll get back started for the last couple slides for this session.
One of my colleagues is a microbial ecologist. He got into like a Twitter fight with, I think like somebody runs some Twitter account in Lake Superior. Yeah, I don't. Where in Michigan are you from? You guys too?
I'm from Rutgers. Veterinary lab. I do deep ocean volcanoes, volcanic systems. I've run into him, but I haven't interacted with him much. He was on the committee of someone's dissertation I was watching.
But I run into him at various micro things.
Yeah, what was her name? I don't remember the name of this. She was one of his microbiologists. Basically, she was looking at contaminated groundwater somewhere in the Appalachians. She found some organisms.
What do you do at Michigan State?
You have to plan it more nutrition than food science.
I'm not yet.
The upside of being associated with the department that is that you get to see like when they're doing like, trials or things like taste tests and yeah. I see. There's a department like that in Minnesota on the same campus, which is where things. We're not real close to them, but every now and again I'll see flyers for like, various tasting.
So we can do this last little bit here
Yeah.
to finish off the morning session. I did realize in my brain, I'm going, well, we're getting really done really early about this. Like, what's happening here? What's going on? And I realized, small group, we have like a really long introduction part. Everybody stands up, says a little thing about themselves. So what we're going to do is after the Unix session is done, before we go to lunch, so you don't have a 2-hour lunch, we'll do a quick rundown around the room, and then you can stand up and say something maybe about your research. And then people can find people they want to go to lunch with because you stood up and say, I do hydrothermal vents. And everybody goes, I want to go to that person with the hydrothermal vent stuff. Anyway, so that's what we'll do after this. And then we'll break for lunch. So let's just wrap out this is about another 12 slides or so to go. All right. So what I want to show you right now is an actual tool that's going to work on the command line, like everything else that we were just doing. But this isn't just like a basic element of Unix or Linux, right? So you don't have this by default on your Mac. You don't have it by default on your Linux computer. This is something you would have to install. But it's really useful for working on the command line with sequences and sequence files. So it's something that I wanted to show off and show people how to work with. So it's called C-DEBI. The link is here in the presentation. It should also be in the install list. And so what C-DEBI allows us to do is allows us to work and manipulate sequences in a way that make a lot of sense. Of like, oh, I wish I could do something like this with a really big sequence file that I have. And generally C-DEBI allows you to do something like that, whatever this is. So there's a couple things inside C-DEBI that you can work with, right? So C-DEBI is the name of the program, and then it has 3 major avenues of information you can work with, plus a couple of extra ones on top of that we probably won't cover. C-DEBI convert is a command that will take a file and modify it and create a new file. C-DEBI modify will take a file and make changes to that file itself and then not make a new file, right? So you end up editing within the file instead of creating a new one. And then info, which I find really useful, which allows you to take all of the information about files and of FASTA files in a folder and put them and print out some information about it. There's also these other elements. So you can back translate a line, you can extract IDs, you can quality filter and primer trim within C-DEBI, but that's not something we're going to cover. We're actually going to cover some of those topics in the afternoon's first session. So what can we do with this? All right. What we can do is we can use the file IDs that we just generated to pull the actual sequences associated with those IDs out of our files, right? So we can do something like this, where we can take C-DEBI, like that, convert, and then it's going to be -- include from file, like that, new clean IDs. And so this was the one that we were just working with, right, where we took all of the IDs that we had and we removed the caret from it, because this program would not accept a list with carets on it. That's not what it's looking for. It's just looking for the ID names. And then what we put in is we put in new clean IDs, right? So then we're going to put in the name of the file that we're pulling these IDs from, which is group12_contigs.fasta, a space, and then the name of the file that we want to generate and put it into. So in this case, we're going to call it new group12, _contigs.fasta. Okay, so we hit enter. That should work. If it doesn't work in your first try, there's a good chance that there's a typo. That's a lot to type out. It's really useful if you're using the tab to autocomplete. That means you're not going to make a typo error when you're typing out group 12 or something like that, or new clean IDs. Did everybody get that to work? Okay. All right. So let's jog our memory about what's in new clean IDs. So if we take less and look at new clean IDs and look through that. So that's got all of our 47s. It's got all of our 51s. It doesn't have duplicates of the 47s, right? And so if we wanted to know, well, how many IDs are in there in new clean IDs, we can do word count new clean IDs. So there are 18 new clean IDs. So if we do the exact same thing and ask C-DEBI extract IDs, right? So this is going to be very similar to if we were doing grep and using the caret. You can do C-DEBI extract IDs, new group 12. Whoop. Like that. So this is going to extract IDs, and we can ask, well, how many IDs are in there? We hit enter, and there's 7. Okay. this is something that we're actually expecting, because if we remember, we got all the 47 IDs from group 12 contigs fastas way back when we were working on this. And we got all the 51s from group 20. And so group 20 has a whole bunch of different contigs than group 12 does. And so when we asked SeqMagic to go through these IDs, find the matches, pull out the sequences that it matches to, we only wanted to find the ones that it actually originally came from, right? There should be no, not that there aren't numbers that have 51 in it from group 12, but they don't have the same number, like contig full name, as the ones that are in group 20. So what we've done is we created a list of IDs that came from 2 separate files, right? And we've cleaned them up and we've done some stuff with them. And then when we asked SeqMagic to go back to the original files and pull out the corresponding matching IDs, it just found the ones from the source file to begin with, right? So if we did the same thing and did SeqMagic convert include from file and then use group 20 as our starting point, we'd pull out and have a new group 20 that just targeted the ones that had 51, right? So we've been, this is a way you can make a long list with lots of IDs in it and then use that to search through multiple different FASTA files if you wanted to. It's, one of those things where, when you can do it, you can start thinking about ways of how you might be able to manipulate files in a way that is useful to you that I can't predict right now what you actually want to do with them, right? You have a list, right? You've gone through and you've spent some time and you found all of the sequences that do a certain thing in 7 different files. But now you want to put them all together, right? So you make one long list of the IDs that you want to find and systematically go through and pull out the sequences. And then you can do something like concatenate them at the end and make a big file of the ones that you're actually targeting, right? So the idea here is that we're giving you pieces that allow you to move forward and work with the data that you have, right? And so the next thing we can do is we can use the info command, right? So if we look at what we have here, we have a bunch of different FASTA files, a bunch of different ID files. So by default, well, not by default, but what we're going to tell it is we're going to use SeqMagic info and look into all of the files that have FASTA at the end of it, right? So not the ID files, nothing like that. So we can do SeqMagic info star, right? Our star allows us to, it's a wildcard, allows us to find anything that has FASTA at the end. And anything before it, it can actually look at and determine and say, oh, here's FASTA at the end. I want this file, and I want this file, and I want this file, and I want this file. So the 4 FASTA files that we have should be listed here. If we hit enter, it should generate a little table like this, where we have the name over here. It asks a question of the file is in an alignment. If in this case, none of these are alignments, so it's all false. And then gives us some information about what the shortest length of the sequence is in the file, what the longest length, what the average length is, and how many sequences are in there, right? And so, before when we did the word count, we saw there was 132 and 12. That's great. We saw there was 203 and 20. And then in our new file, which we just looked at, has 7 because we pulled out just the ones that have a 47 in it. All right. So we can actually put this all into a file to keep it safe, right? So we can do SeqMagic, we can hit up, SeqMagic info star FASTA, and we can put a little caret here, and we can call it FASTA info, just like that. Hit enter, and so now we should have a new file. It's gonna, if you look at it, if we do less FASTA info, it should have that little table that was printed to the screen is now stored in there. And now we can manipulate this pseudo table, right, which just is using tabs as a way of keeping the data separate from each other. We can actually pull out certain aspects of that using the cut command, which is another Unix staple. So cut allows us to target specific columns from that output, right? And so if we do cut -F, where F here delineates the field that you want, and field refers to columns starting at 1. So the first column is 1, second column is 2. We can do F, we can put in a number. So in this case would be 2. And we can put in FASTA info. And now if we hit enter, it's just showing us that second column. And so we can hit up and we can play with this a little bit. And so we can do F 2, comma, 4. And so the comma is going to tell it print out the second column and the fourth column. So you get something that looks like this, max length with the alignment part. And then if you go back and hit up again and you change the comma to a dash, it's going to print out all columns between 2 and 4. So you get 2, 3, and 4. And so this is, I mean, this is one of those, another command where you can be using this a ton of times, all the time, if you want to be only grabbing out certain information. So you can use grep to find all the lines that have a match that you want for a tab delimited file. You can find all the lines you want, and then you say, oh, I don't want all of the columns. I just need the last 3 columns. And then you can put whatever those numbers are and pull out that information. It's a way of modifying the data that you're working with to make it simpler for you to look at, for a program to look at, however you might be working with it, right? So cut allows us to do that. So right now, cut is working fine because by default, it uses tabs as a delimiter. And FASTA info file has tabs in it, which is really great. But you can do lots of other things, right? We all have seen before, even if we've never used them before, comma separated files that come out of an Excel document. And so we can change that. I'm just going to give you an example. We can change that. We can do cut -d, and then we can tell it that what we're looking for. So the delimiter for what is actually separating items is not a tab here, but it's a comma. I don't have a comma separated file to give you that example, but you can do that. And you can get, you know, people get creative with the separators that you use. So sometimes people will use the pipe as a separator. This allows you to break up things however you want, however somebody gives you the information. There's always, you know, the ongoing complaint of the people who store their files, especially, you know, more back in time that you go, who store their files as comma separated files, and there's no way to get at them. There are ways to get at them. Unix will allow you to do that. You can do things with it. You know, they're not lost forever. Sometimes they are, not now. That's not what we're talking about. All right, so some additional commands that you can work through to just give you an idea of what we have. And so history might be one of these things that's really overwhelming because generally it will keep, so I use the virtual machine and I make it and send it to you guys, and there's a lot of experimenting that goes along with that. So, and sometimes it will have stored everything that I've ever done in the virtual machine ever. So this number just keeps getting bigger and bigger. But if you type history, so if you're trying to think back earlier in the day, you've been working on the command line all day, you can't remember what you did, something worked great in the morning, but it's 4 o'clock in the afternoon and you can't remember what it was, you can type history, and this will print out, oh good, it's only the last couple stuff. It will print out a full working list of what you've done in the history of the commands that you've run, right? So you can scroll back up and we can see everything that we've done today, and you can get an idea of the order that we worked it in, and this will keep going back up and go back up and through time. And like I said, I think 2,000 lines, so this probably goes back to 2016 when, the virtual machine was for the EcoGeo version of it. So this is something that I just want to bring to your attention. It's not something that we're going to work with, but if you ever run into a problem. So one of the things that programs will ask about is figuring out where your path is. And your path is something that's generally, there's a path for the whole computer being used, and there's also a path for each user on a computer. And a path is where information is stored, where the computer looks for first when you're trying to run commands and look for data, right? So your path, knowing what your path is can be useful for some programs going forward. So if you hit echo $path, it lists all the directories, right? So there's only one user on this virtual machine, so there's not a bunch of extra information, but this is all of the places that the computer looks for commands, right? So like seek magic, right, is one of those things where we can tab it out. It's stored in the computer, and to find seek magic, it looks in all of these directories first before it says I can't find seek magic. And so this is just something, this is one of those things of like 3 weeks from now, 2 years from now, if somebody goes, what's your path? I don't remember what a path is. And you can come back to this one slide at the end of the presentation and be like, oh, that's how I figure out what my path is. It's not something you're going to deal with on a regular basis, but it's just something to be aware of. And then this last one here on this slide is nano. And so this is not something I highly recommend doing. You should be able to, on your computers locally, if you're, working on your laptop and sending things to a server, you want to be working in some text, manipulating document, right? Text editor is really good because it doesn't have the weird command returns that happen in Windows files. There's a couple other text editors that this also works really good for. But say you're on a server and you need to get into the information that's there, but you don't want to pull everything back down. You can use Nano to actually do text editing within the window of the command line, right? So if we look here, let's pick one that we can work with. We can work with one of these temp files. So we can hit Nano temp1. So this is going to look very much like less, but now we can make edits to it. So we have a little cursor. We can go around. I can change config to sequence here. I can get rid of that. I can make this a 5. I can make this a 23. And there's a bunch of different commands down here that allow you to get out of it or to be useful, right? So this command W, if I do that, so that for I think all the computers should work. It should be your control button that does this. So if you hit Control W, it brings up a little search bar, and I can type in 457. And if I hit Enter, it'll take the cursor to that point in the file, which is pretty useful. If this was much longer, I could page up or page down very quickly using Command Y or Command V. And then when I'm done with what I'm working here, I'm going to hit Control X. That's going to ask if I want to exit. And so right now, nothing's been saved. So if I say yes here with a Y, it'll save all the changes. If I save no, it won't save any changes. So I'm going to say yes. If this is what, this comes up, it's asking which file do you want to write this to. So right now we're in temp1. So if I wanted to save it within temp1, I hit enter. If I want to make a change, I can change the name here. I can make it temp12 or temp25 or whatever. And then you hit enter, and then it's made the changes. And now if you look at less temp1, there's all the changes we made, right? Sequence is now there. The numbers have changed. Not that I remember what the original numbers were, but they are different. So you can do that. So that allows you a quick shortcut if you're working on the command line where you can go into a text file and make changes there. I'm not saying that's how you should be doing it. I'm saying that's an option for when you need to do it. Right, okay, so that's just the Nano example. All right, and so then the last thing that we want to do and play around with a little bit is this idea of a bash script, even though it doesn't say script here. Look at that, on the fly fixing. So what this is going to do is, and what we want to do is we want to, there are lots of times we're going to do stuff that gets pretty repetitive on a command line. We don't want to do it over and over and over and over and over and over again. And there are commands that you'll want to run, and they'll run for a while, and they'll finish in the middle of the night, and you don't want to have to wake up and start the next one right away. So what you can do is you can actually make a text document that has a list of commands in it and change the permissions on it to make it go from a text document to something that's executable, something that the computer can actually run. So if we go less and look in simple bash script, there's a couple seek magic commands that are in there. And so what this is going to do is it's going to run seek magic convert. The little screen's in the way. I'm going to move that for a second. So it's going to run seek magic convert. We're going to use new clean IDs to pull out the corresponding correct files from their original sources. We're going to output a file called bash result1, bash result2, and then we're going to rerun the seek magic info.fasta command, right? So we get Q to get out of that. Guess I'm plugged into the sound. And so right now, so it's actually already been done. Right now, simple bash script is executable. You can tell that by the green. This is not something that is common on all terminal windows, right? So sometimes you'll get these nice fun colors to tell you if something's a regular plain text file. Sometimes you'll get stuff that will tell you, if it's a directory or not. We were seeing directories as blue before. So what we can do is we can look at, if we do ls-l and hit enter, it should come up here at the bottom. So over here in this left column right here is all this information about what these particular files can do. And we can get into more details if you have questions about this. But what the R's and the W's represent are reading and writing, right? So you can see most of these files, multiple IDs, clean IDs, they have this read and write and read and write and read setup so that you can look. So these are just text files, something that you can read and you can write to. Simple bash script right now has these X's, and these X's represent the ability to execute, right? And so the way we do that, and so if this wasn't the case, we can run the command chmod, which changes the file modes, right? And so we can do chmod 775, which is the way that you actually tell it that you want it to be executable, and you type simple bash script, and that will change it again. So originally it was like, so don't follow along here for half a second. Right, so if I do that, simple bash script is no longer executable. It's just a white right there. So chmod, and if I look at it here, you can see new bash script is right here, and the X's have disappeared. So if I do chmod 775 simple bash script ls-l, you can see not only has it returned to being green, but now there are X's that make it executable again. So that's really nice, right? So what it means is now we can actually run the commands that we've saved in there in the order that they're printed in the text file. So we can do that by hitting / simple bash script like that. And if we hit enter, it's going to be a slight pause, and it's going to run the things that we asked it to do. And we know it's going to has done that because now in the results, we have these bash results 1 and 2 in the output that weren't there originally before it started, right? So it ran through that. And if you want to see what happens if we do something like this, right? So again, simple bash script's the only one. If we do / temp1 _ IDs, it's going to yell at you and say permission denied. And that's because it's right now it's only set as a read write file. All right, and so this is the last slide. I touched on this at the beginning of the day. Again, if you have the need to start working with lots of data, you need to find a high-powered server to work on, because this is not going to work on your laptop, unfortunately. Does anybody have any questions? Are there any questions from the webinar? We can answer those now. Anybody have any questions right here? Otherwise, we're going to close the webinar down. We're going to start back up again at 1 o'clock Pacific time. Here, we're going to do a fun introduction session for everybody so we can all get to know each other, and then we're going to go to lunch. Does anybody have any questions? Wait for the webinar people. Going once, going twice. Sold. All right. Well, webinar people, we'll see you back here in 2 hours. Everybody else, let's start talking with each other. Stop sharing.